Okay, so we are partway through carbohydrate lecture 3, right? Went through all this great stuff from biochemistry and regulation. This is where we are, right? So one of the things that you'll see as we go through this and the study guide, you'll see that there's the emphasis in here is going to be regulation of glycolysis and mostly on the upper part of glycolysis. So I'll have on the study guide which of these reactions you need to be able to draw the structures. I think I mentioned most of them last time, but we can also see that the 2 points of regulation that we're going to focus on are step 1 and step 3. So we're going to go back through the enzymes. So you'll obviously need to know the enzymes, their specific reactions, and how they're regulated. And then as part of that, we'll look at, for example, how it's regulated on a cellular level, but also in the system by hormones. So we'll talk about all of that. So down here, essentially, you don't have to know any of these structures on the bottom part of glycolysis. But you'll see you need to know the structure of pyruvate. And so we'll go through that. The other reason I like this diagram is it shows where galactose enters and fructose. So we're going to talk about why that makes their metabolism unique and different than glucose. Okay? Good on this? So here's step 1. This is done by hexokinase or glucokinase, right? Remember, it depends on the tissue. And this is just glucose to glucose 6-phosphate. So you'll be able to draw the structure of glucose and the structure of glucose 6-phosphate. And all you have to do is put a little line and then put the P for phosphate. That's fine. And then this one, the second key point is a reaction that's reaction number 3. That's fructose 6 to fructose 1,6-bisphosphate. And that's done by an enzyme called phosphofructokinase 1, PFK1. And we'll talk about its regulation. This one's more important. PFK1 regulation, most important point of glycolysis. So we'll spend some time on that. Now, we've talked about some of these differences before, differences between glucokinase and hexokinase. And one of the things we said was, well, they're expressed in different tissues. And we also said that hexokinase has feedback inhibition. That's by glucose 6-phosphate. And we also said that hexokinase has a lower Km than glucokinase. And I'll show you why that's interesting in a minute. And then glucokinase, we said expressed in the liver and you guys don't have to hold back. I know you guys go through your notes after every lecture, and you know it's on the tip of your tongue and you don't want to show off. Beta cells. Right? It's not negatively regulated by glucose 6-phosphate, no feedback, and it has a higher Km than hexokinase. So let's look at this really quickly. Right? Do I need to say anything more? Is this all just self-explanatory? You've seen these curves, you're like, oh, that's a Km curve. Right? Remember this? And you go, oh, okay. I'm going to set up a reaction, and I'm going to do kinetics on an enzyme, and I'm going to say, most of the things in this, let's say in this reaction tube, are constant. 2 things that change are substrate concentration. So in other words, I'm going to set up this reaction in a tube, and this is automated. I'm going like this, but at this point it can be automated in labs, right? And now I'm going to add more substrate to this concentration. And as I add more substrate, I get more product. So this is the appearance of product. So what's over here? Yeah, here's rate or velocity. Is how fast my product is appearing. So essentially, as you're adding more substrate, and then it goes like this for a while. It's basically kind of it's exponential for a while, then it starts flattening off. All right, what's going on when it's flattening off?
Enzymes get saturated.
All right, we go. All right, so as this is happening, and as your substrate and enzyme combine, you can basically saturate your enzyme. And that's why it starts falling off. So remember, your enzyme concentration stays the same, but you're adding more substrate. So the tighter that substrate binds and forms this complex, the more it'll slow down. And that's affinity. That's a simple way of looking at affinity. So if an enzyme has a high affinity, it tends to be slower. And you'll see how all that works in 1 second. All right? So now what we do is we go, okay, well, this enzyme, I'm going to leave the enzyme concentration the same, but as I add substrate, you can see the velocity of this reaction is falling off as the enzyme becomes saturated, right? And this eventually becomes kind of flat here. So what do? Well, this is as fast as it can work. So we're going to say this is maximum velocity. And then you guys know this, right? So here's my Vmax. So now what do? Now you go, okay, what's half my Vmax? So what's this? That's the Km. Right? Let's add some flavor to it, some specifics. We're talking about glucokinase and hexokinase. What's our substrate? This is glucose concentration. And our product is glucose 6-phosphate. So how fast can each of these enzymes convert glucose to glucose 6-phosphate? Well, let's look at glucokinase first. We said it's got a large Km, and that Km is 10 millimolar. Again, who cares? What does that number mean? Let's compare it to real life. If this is 10 millimolar, and if we say, well, that's the Km, in other words, right here, right? 50% velocity, 10 millimolar, that's how they came up with that. This is really important. It says normal fasting blood sugar level. You can tell this is from a biochemistry text. This says sugar. We would say normal fasting blood glucose level. Look where it is. About 4 millimolar. That means fasting, that's a typical blood glucose concentration. So we know that must be, well, that's probably about 90 mg per deciliter if you convert it. Because you guys know, you guys remember from last time, 90 to 110 mg per deciliter is fasting blood glucose. So that's where we are. This is fasting blood glucose. So in other words, in fasting scenarios, this enzyme, it's only working about 30% of its Vmax. And where is glucokinase? In the liver and beta cells. So in fasting scenarios, that enzyme is not even working anywhere near its maximum potential. So it has no problem converting glucose to glucose 6-phosphate at that fasting blood glucose. Okay? So now, let's say I consume a carbohydrate meal. Let's say it has 100 grams of carbohydrate in it. It's very reasonable that this value, fasting blood glucose, is going to change from about 90 to, let's say, 150 or 200. It's very common for it to double. Okay? So let's say it's 10. It went from 4 to 10. Even after a meal, when blood glucose goes up significantly, how fast is glucokinase working relative to its Vmax? Half. In other words, even as blood glucose goes up to that high level, glucokinase is nowhere near saturated. It can continue to make glucose 6-phosphate without being inhibited, no problem. It's not really falling off at all yet. So what does that mean? That means after a meal, glucokinase in liver cells and beta cells can convert significant amounts of glucose to glucose 6-phosphate. So that means after a meal, the liver can phosphorylate significant amounts of glucose 6-phosphate. And remember we said the significance of that? It traps it in the liver. And as I'll go through the other one in a second, you'll realize what the difference is. Okay? Fast enzyme, high Km, low affinity. Now look at hexokinase. That's the blue one. Look at its Km. 0.1. What's the fold difference? 100, right? It's a 100-fold difference. That's a big difference in the enzyme. Why would our body do that? Because now look what happens. Fasting scenario here. How fast is hexokinase working relative to its maximum velocity? 90%, about. Which means at that concentration, it is effective at converting glucose to glucose 6-phosphate. It's doing that at a high efficiency, but it's close to its maximum rate. And why is that? Because it has a low Km but a high affinity. That means even if glucose concentrations are low, that enzyme can still phosphorylate it to glucose 6-phosphate. In other words, even in fasting, that enzyme works pretty well. And why would that be important? Where's hexokinase predominantly? Muscle. So even in fasting situations, muscle fibers can phosphorylate and trap glucose 6-phosphate. That means even in fasting, they can get energy from glucose. Now, let's say after meal. Now, let's say we go up to at least 10. What happens to the enzyme? It doesn't work much faster. So that means after a meal, which tissue has more of an advantage of phosphorylating glucose? The liver. And why is that important? Because the liver can do what? The liver can store it as glycogen and release it later as glucose. See why that's important? Sure. So after a meal, the liver has the ability to phosphorylate and trap glucose as glycogen, so it can be released later as glucose.
So is like the glucokinase, like after the meal, is like the glucokinase is going to work more than the hexokinase?
It'll make more product. Absolutely. Yeah. Has more capacity.
Then it's opposite when you're fasting.
Yeah. And if you think about it, in fasting, why doesn't the liver have to do that? Why doesn't it have to be able to phosphorylate glucose effectively at low concentrations? It doesn't need to. It can make glucose, and it has glucose stored. So that one might take a little bit for you to kind of think about a few times and get it to click, but that's why those enzymes have different characteristics and they're on different tissues. It's all about what the liver does during fasting. Right? It can store glucose and then release it as necessary in between meals or overnight, for example. Okay? I'll let you think about that. If you have questions about that as we go through, but that, to me, this is a great example of why the kind of the kinetics and enzymology is so interesting, is because our body uses different forms of enzymes to do the same reactions but with different characteristics. And that's one thing that significantly affects our metabolism. Okay? So now let's talk about PFK1. So remember, now we're at step 3. We're going from fructose 6 to fructose 1,6-bisphosphate. Single most important regulatory mechanism of glycolysis. This is the first committed step in glycolysis. This is why we say it. This is why I like this diagram too. You guys erased this, right? Hexosmonophosphate shunt. You cross that out. What did you write right here? Yay, no. What did you write right there? Glycogen. You should have glycogen in there because glucose 1-phosphate can go to glycogen. Okay, so that's relevant for this one because let's say glucose goes into a cell that's converted to glucose 6-phosphate. I don't know why I'm doing stretching exercises. Right? Have we committed to glycolysis yet? No, not really, because we can go this way, or we can go to glycogen. We can go glucose 1-phosphate, then glycogen. The next step, glucose 6 to fructose 6. Have we committed to glycolysis yet? Not really, because we can go right back this way. But once we go here, see, that's unidirectional. So once we go here, we're committed to glycolysis. That's what that means. So this is a key regulatory point. Once we push the fructose 1,6-bisphosphate, we're moving towards pyruvate. So that's kind of big picture where we are. So let's talk about how this is regulated. This is out of a biochemistry text, and this is allosteric regulation of PFK1. So in other words, PFK1, allosteric enzyme, which means it's got an active site and allosteric site. So here's our reaction, fructose 6-phosphate to fructose 1,6-bisphosphate. Here's the enzyme. So please say, and practice spelling, phosphofructokinase 1, because there is a phosphofructokinase 2. PFK1 is the one that does this reaction. And this has 4 regulatory molecules. So these are allosteric modulators. Number 1, ATP. That inhibits PFK1 activity through an allosteric mechanism. Does that make sense? So you can memorize these, or you can go, well, what should ATP do? Increase or decrease glycolytic activity? Who says decrease? Who says increase? Okay, 3-0. Great. Good poll turnout. Right, so if a cell already has significant amounts of ATP, we can slow down glycolysis. We don't need to make more. So ATP, and where's most ATP made in the cell? In the matrix. That ATP can leave the matrix and downregulate glycolysis. We'll look kind of big picture in a second. Citrate inhibits. Where's citrate from? Krebs cycle. So if Krebs cycle has been active and citrate is at a high concentration, some of that citrate can leave the matrix, bind PFK1, slow down glycolysis, essentially decrease more citrate production. So these are both kind of feedback, right? It's kind of cool, right? The cell is regulating energy production based on how much energy it has. ADP, that makes sense, right? If you have a lot of ADP, that means you've expended a lot of energy and you need to make more. So if ADP concentrations go up, PFK1 activity stimulated. So you can imagine this. Let's say here's PFK1. Let's say this is our active site. So let's say this is fructose 6-phosphate, ready to convert to fructose 1,6-bisphosphate. Here's our allosteric site. These are allosteric modulators. In some scenarios, they're competing for the active site. ATP and ADP can both bind it. Depends on which is in a higher concentration. So if we have a lot of ATP, it's probably going to bind before ADP does. So they would bind to this site, downregulate activity. And why can each of these modulators have a different effect? Because their structures are different. ATP is different than ADP. The enzyme senses that. So let's say ATP is bound here. That enzyme is going to shift to a less active form. Right? All these molecules can do that. And same thing with citrate. Downregulates. And the next one is a little bit different. Fructose 2,6-bisphosphate is probably the most potent effector of PFK1, and it stimulates activity of PFK1. And if you look at that reaction, where does fructose 2,6-bisphosphate come from? Fructose 6. Right? We call this enzyme PFK1 because we're phosphorylating carbon 1. So it's kinase 1. The difference in this molecule to this one is this molecule has a phosphate on carbon number 2. So what would we call this enzyme? PFK2 does this. So here's where it's a little bit different than the other ones. Let's imagine glycolysis is starting to go fast and fructose 6-phosphate is beginning to accumulate. This enzyme has a way of feeding forward. So as fructose 6-phosphate goes up, some of it becomes fructose 2,6-bisphosphate. And that stimulates PFK1 activity. Let me say that again, because I got nothing. Okay. As fructose 6-phosphate goes up, some of it becomes fructose 2,6-bisphosphate. And that allosterically stimulates PFK1.
So why, if there's already a lot of it, it seems like if you have a lot of it, wouldn't you want to start regulating down the opposite way? Or it's like you have a lot of it and now having a lot of it is stimulating more of it.
So you're also relying on the other feedback mechanisms. So in this case, it's basically saying there's a lot of substrate for this enzyme. Unless other molecules are slowing down the enzyme, we're going to push its substrate through the product. Now, as that goes through glycolysis, if a lot of this now goes through glycolysis to pyruvate acetyl-CoA and goes the Krebs cycle, makes ATP or citrate, now it's going to start feeding back. So that's why I say it's one of the most important and potent ones, because it doesn't allow fructose 6-phosphate to build up. It pushes it forward and then allows other feedback mechanisms, if necessary, to decrease the speed. Yes?
So could you say, so is fructose 2,6-bisphosphate like an intermediate? Or is it required in order for PFK to work?
No, it's not required. It's actually its major function, allosteric regulator.
And that, like, the function of that would be so fructose 6-phosphate doesn't build up?
Correct. Yeah. So it can convert. Uh-huh.
So does the fructose 2,6-bisphosphate, is that not part of glycolysis at all?
Correct.
It's only made as an allosteric?
It might have other metabolites, but it's not itself a glycolysis molecule. Yeah. Matter of fact, I'm sure I'll have to look at the poster in my office. to see all the potential molecules for that, but it's not glycolysis. Yeah. Good? Okay. So that's cellular. Okay, that's how energy levels in a cell are regulating energy production. PFK1 is important for that. So now let's look big picture. Let's talk about hormones. This is hormonal regulation of PFK1. Insulin increases the activity of PFK1. Due to increased, what does that mean? Enzyme concentration. That means insulin makes more PFK1 protein. Does that make sense? When's insulin up? When glucose is up. So blood glucose goes up, insulin's released, insulin helps cells upregulate glycolysis. Makes sense, right? Now, its counterpart, I'm not going to say, Darth Vader or whatever. The other side of this, glucagon. Sorry, I grew up like Star Wars was my childhood. I'm like, wow, it just blew me away. It was easy. I don't remember how old I was, but I was easily blown away. But it's amazing it's still around. Here's glucagon, the opposite. Remember, glucagon does the opposite. So when blood glucose is low, glucagon goes up. Makes sense, right? If glucose is low, that means there's not a lot of glucose available for glycolysis. So downregulate glycolysis. And glucagon does that. So as glucagon goes up, it decreases PFK1 activity, mostly by decreasing fructose 2,6-bisphosphate. Glucagon decreases PFK1 activity mostly by decreasing fructose 2,6-bisphosphate. Right? It removes that stimulating modulator. As glucagon goes up, it decreases PFK1 activity, mostly by decreasing fructose 2,6-bisphosphate. I think I got that right. Right, so that's kind of the big picture or systemic regulation of PFK1. Remember, those hormones are binding cellular cell surface receptors and then causing signal transduction that regulates PFK1 inside cells. So we've looked at cellular and hormonal regulation of PFK1. Now, oops, that was fast. So now if we go glucose through glycolysis to pyruvate, what are the options for pyruvate? Under anaerobic conditions, what does it become? Lactate. Also known as lactic acid. Basically the same thing. Under aerobic conditions, what happens? It can become acetyl-CoA. And where does that happen? Matrix of mitochondria. We'll talk more details in a minute. Okay? And here's another option. What kind of molecule is alanine? That's an amino acid. So this is a very direct relationship between amino acid metabolism and carbohydrate metabolism, right? So some pyruvate can be used to make alanine. And alanine can be converted to other amino acids. We'll talk about that in a few weeks. So those carbons from glucose are essentially supporting amino acid metabolism. Based on this, do you think alanine is an essential or non-essential amino acid? Non-essential, pretty easily made. Basically, if we have this, if we have an amine group, pretty easy to convert that. Okay? So far, so good. In a few minutes, or in a little while, we'll look at this backwards for gluconeogenesis.
Would it create alanine under both anaerobic and aerobic conditions, or only one?
Both. Could, yeah. Fructose. A lot of bad publicity on fructose, right? Stay away from fructose. Is fructose bad for us? I mentioned this a little bit before, right? Where we say, I wouldn't say it's bad for us, but too much of it can be harmful. And we'll talk about how. So if we look at this, first of all, what are significant sources of, let's say, glucose in our diet? What's going to hopefully or tend to be the most significant source of glucose in our diet? Starch. Probably. How about fructose? Name some significant dietary sources of fructose. Fruit. Nutrition class. Fruit. What else? Sucrose. High fructose corn syrup. Anything else? Honey. Can be. So would you avoid those foods? Let's talk about that. Look where it comes in. Again, this is one focus on this slide, not your textbook. The textbook has a step missing. Here's fructose. Now this is minor and major. As a matter of fact, don't worry about the minor. Remember we said about 100% of fructose goes into the liver. So we're going to look at this step. And I probably drew this not very good. I added this in Photoshop, sorry. This fructokinase is inside the cell. Fructokinase is doing this. Fructose to fructose 1-phosphate. So fructokinase is in the liver, and it converts fructose to fructose 1-phosphate. Well, how did it get into the cell? How did it get into the cell? Is a liver cell. GLUT2. There's 4 characteristics of GLUT2 that make it optimal to be on the surface of liver cells. One of them transports fructose and galactose. So that's GLUT2. Goes in, phosphorylated by fructose kinase to become fructose 1-phosphate. Are we in glycolysis? No. Fructose 1-phosphate becomes glyceraldehyde and DHAP. In other words, we got 1-phosphate here. When we split that fructose molecule, we end up with 1 molecule, GHAP, that has the phosphate. Is this glycolysis? Yes. Is glyceraldehyde? No. It needs to be converted to G3P using a second ATP. Now, all of those carbons from fructose in glycolysis. So can fructose metabolites enter glycolysis? Absolutely. That's where it goes. But it's got a slightly different route. Right? It doesn't become glucose 6-phosphate. We're below that. I erased some of the steps on the early steps of glycolysis. We're fructose 6-phosphate. Fructose comes in below that. We're in the latter stages. How many ATP were invested to get there? 2. Is that the same as glucose? Yeah. And then it goes through the latter steps. And so how many ATP do we get per fructose molecule? Plus 2. Right? It's the same as glycolysis. We get the same energy production. We invest the same energy from fructose. But what's different? We came in here. We came in below the major regulatory point of glycolysis, PFK1. So what does that mean? Yes, it's regulated differently and sometimes not very well. In other words, an excess of fructose can be a problem. Because now look where we are. Let's say, for example, let's say in a young individual, a kid, and let's say this kid is not being physically active enough, and they're consuming significant amounts of soda or something or candy with a lot of fructose. Now you've got a scenario where the kid is taking in high calories and a lot of it's fructose. That fructose can go into the liver. In that situation, we have a lot of ATP, a lot of citrate, because energy production is high. But fructose is not being pushed away from glycolysis. It continues to go into glycolysis. So it will continue to run to pyruvate, to acetyl-CoA, to citrate, to fatty acid production. And that's when it can be problematic. Too many calories and too much fructose can stimulate hepatic lipogenesis. High calories, high fructose can stimulate hepatic lipogenesis. High calories and high fructose can stimulate hepatic lipogenesis. And remember before when we said, let's say this becomes citrate. And then if we have significant ATP, we'll look at this in a second, this moving forward can be inhibited. And so the citrate can go into the cytosol. And citrate can convert back to acetyl-CoA, to fatty acids, to a fatty acid, to triglyceride, to VLDL. And this triglyceride can go to fat cells. And that's one of the problems. It stimulates lipid production if it's chronic like that. So that's something that we have to watch, excess levels of it. Okay? And so I said the other time, I said it can, too much fructose can inhibit leptin signaling. Remember we said that before? Leptin? Ring any bell? Okay, we talked about that. And then we also said hepatic lipogenesis. So now that kid doesn't get the satiety effect that they should. And they can continue to repeat that cycle by consuming too many calories. So it also happens in adults, but it's even more problematic in someone that's smaller like that. Good there? So that's why it's different. So an adequate amount of fructose is fine. Excess is a problem. And now let's compare that to galactose. Now here's galactose. And if we, again, go back to this, here's galactose, becoming galactose 1-phosphate, and then glucose 1-phosphate. See what it did? Converted from galactose to glucose. And now, glucose 1-phosphate can either go to glucose 6-phosphate or glycogen. But see where we are? We're above PFK1. So galactose tends to be better for things like hepatic glycogen synthesis. Here's our steps. And the enzyme is galactokinase. So it's different kinase. makes this, converts to this. Now this can become glycogen, or glucose 1-phosphate can become glucose 6-phosphate, go through glycolysis, or glucose 6-phosphate. So here it says glucose 6-phosphate. This should say to glucose to the blood. Sorry about that, right? This doesn't go out. This does. So we'll talk about that. The liver can do that. And so galactose metabolism is a lot different than fructose. What are dietary sources of galactose? Probably the main one, dairy in lactose. Good there? Okay. So let's take a break. I'll stop this, and then we'll continue on with carb lecture 4.